Alrighty, guys, and welcome to another round of virtual slides. So we're going to be going through some blood spatter information as well as learning a little bit about how blood can be used to help put together the sequence of events at a crime scene. So the first half of this slideshow we're going to do in class when we do the blood typing lab, and then the second half is what you're doing right now, what you'll be working on at home. So let's go ahead and get started here. So just for copyright sake, I have this video linked on the videos in the notes folder. I'll have that connected to this video assignment here, but please watch this video. It's very, very well done. It goes through a lot of different information to know about blood spatter. So like I said, I can't show it in this video, otherwise I'll be copyrighted, but make sure that you do click on that link so that you can get this information because it's very good. We've seen this guy a few times beforehand. All right, so now that you've watched that video, hopefully, you know a little bit about blood spatter, how it's created, what we look for when we're analyzing it. So now let's break down those details. So essentially, when we're looking at blood spatter, there's quite a few different things that go into play when it comes to actually creating that pattern. So when a wound is inflicted and blood leaves the body, a blood spatter pattern might be created. And this pattern can then help reconstruct the sequence of events. Looking at this pattern, we can look at angle of impact, we can look at the elongation, we can look at the direction, we can look at the common direction. So there's a lot of different parts of information we can get from blood spatter. So there are four main forces that are controlling the blood droplets themselves, and these all then, of course, lend into the creation of the blood spatter pattern. So they provide a lot of information regarding the direction that blood was traveling, the angle of impact, the point of origin of blood, sometimes can even help us determine the manner of death. And once again, this is all just based off of the way that these four forces here work on the blood droplets themselves. So gravity has an impact on the just speed of which the blood will fall. It also can pull blood down a wall to certain effects. Cohesion and adhesion work together to keep the blood droplets together. So cohesion attracts blood molecules together. Essentially, cohesion is a force that has water attracted to itself because blood has so much water in it. It's able to practice the same properties. And then adhesion is also another property of water. While cohesion is water attracted to itself, adhesion is water attracted to other surfaces. So the adhesion attracts the blood to the table, while surface tension prevents the blood from flattening out. Surface tension is a result of both adhesion and cohesion working together. Cohesion keeps all of the molecules together, so they don't want to separate, they don't want to flatten out. Adhesion keeps all of the molecules stuck to the table. So if you can imagine, all of these molecules are trying to stay together and stay to the table. That's going to give them a good surface tension, which means that it's going to prevent the blood droplet from completely flattening and spreading out. Instead, it's going to stay as much as a ball or as close to a circle as it can. That's why in space, when there's no gravity at play, if you ever have seen astronauts in the space system, they will have little water droplets all floating around. That's because naturally, because of the just factors of cohesion and adhesion, water wants to stay in a ball, wants to stay together. And so that's what we see working here with surface tension. So there's a few different parts of our blood spatter anatomy that we will look at in order to get more information about this blood spatter pattern. So if blood is able to overcome the forces of cohesion and adhesion, they might form small secondary droplets known as satellites. And then the edges of the droplets might also form small extensions known as spikes. So you can see some satellites here as well as over here. And then we've got some spikes going on over here. So there are 6 main classifications that we look at when we find a blood spatter pattern. The first is what's known as passive fall. So blood falling at a 90-degree angle will produce circular drops, and secondary satellites will be produced if the surface is textured. I'm so excited to do this lab because we essentially just get to splatter blood on different textured surfaces at different angles, and it'll be so much fun to study all the different pattern analyses. So a second part of the analysis classification is regarding arterial spurts. So your arteries and your veins are a part of your cardiovascular system. Your heart is what pumps the blood through the cardiovascular system. If you were to get cut on an artery, well, your heart's going to still be pumping that blood. So now if that artery is open, your heart is then going to be pumping that blood out of the artery. So arterial spurts are found on walls caused by the pumping action of the heart. And of course, that creates blood pressure. So it's kind of like if you were to have a hose and then you suddenly split the hose open, a big spray of water is going to come out. Same with our blood spraying out of our arteries. Great imagery there. So the third analysis classification regards the different patterns that might be created from different wounds. So for example, splashes and slashes are shaped like exclamation points, and the shape and position can help locate the position of the victim during the attack. The bleeding victim will leave smears of blood as they touch walls or furniture, so you can use that to then establish a sequence of events. Trails of blood as well can be left by a bleeding victim as they change location. So droplets could be round, smeared, spurts, depending on the type of wound that was inflicted. And then finally, pools of blood form around a victim who is bleeding heavily and remains in one place. If you move that victim, then the blood spatter might be able to connect the first and second location if the criminal isn't actually looking for that, if they're not paying attention to it. Obviously, if you have a perpetrator who is paying a little bit more attention to potentially making trails leading from one place to another, you might not be able to find this. So this slide here just has all 6 of those analysis classifications together if you want to be able to see them all at once. So when we look at spatter detail, there's a lot of different spatter patterns that can be formed from the direction of impact, the angle of impact. And looking at these, we can see not only does the diameter change in terms of the height, but also the length of the blood spatter themselves can change based off of the angle of impact. So as we saw in our last slide, a direct above droplet at 90 degrees is going to be completely round. And then as you increase the angle, or I guess decrease the angle depending on which way you look at it, it'll become more and more elongated. So blood droplets with tails or satellites identify the direction from which the blood originated. So basically you want to go long ways. So if this is what you're considering like the point of the blood droplet, you want to go on this axis here, the y-axis, the longer axis. So when we're looking at the spatter details in terms of how spread out all of the droplets are, this is really correlated to the type of injury that had been inflicted. So a fine mist pattern is produced by high velocity impact, such as a gunshot wound, while something that is more so like blunt force trauma, such as a beating with a pipe, would produce a low velocity pattern. If there's any empty spaces in that pattern, for example, if there was just a white gap on the wall here, then that'll tell you that either a person or an object was present that then got moved after the attack. So here's just some more information about the directionality. So if we have a circular drop, angle of impact's 90 degrees, wound type might be passive, dripping, right? No force. If it's elongated, the point of impact appears darker and whiter than the rest of the spatter. And then that's where we can look at then the angle and the point of origin and then help us determine what kind of wound type might have caused those drops. So as blood droplets move away from their source, the blood droplets elongate and they might produce a thinner tail-like pattern that we saw, and the tail points in the direction of the blood's movement. So as I said, the longer axis, so in this case it would be the x-axis is the longer axis, so we would use that to then be able to locate the direction of the blood. So this is blood as it's leaving the body. Notice it's nice and circular because of the adhesion and cohesion. Once it actually hits a surface, then based off of that point of impact, it'll create different patterns. Here's some satellites from then just that overall blood spatter. So smaller satellite droplets might break away from the main drop of blood, as we had seen here. So also here, these satellites appear in front of the moving droplet of blood, and they're not connected to the main drop. So imagine if you have, I like this picture better. So this is your blood, and here's the blood, and it sticks here, but some of it rips off and sticks here because of adhesion, but then because of just the force, another rips off because of momentum, and then it sticks and it sticks. So just think of if this is the initial spot that this blood hit, it's just going to kind of like skipping a stone, if you think about it that way. Skipping rocks on a lake. So as I've mentioned now a few times, looking at the longer axis of the blood, once you get enough of the blood spatterings together, that'll then help you then determine the source of the blood, or the point of impact, the origin. Lots of different words to describe essentially the same thing here, which is where did that injury occur. So by drawing straight lines down the long axis of blood spatter and noting where all of these lines intersect, it'll indicate the lines of convergence. This area of convergence here is the source of the blood. So this is the location where the victim was assaulted in a way that then created the blood spatter. So kind of putting all of this together, I know that it kind of went back and forth there. There's a lot to cover. So for high velocity, we will see droplets that are much smaller, about less than 1 millimeter in diameter. Here's a visual representation of that. Velocity of blood is moving pretty quickly here, hence high velocity. And so some examples of that might be like gunshot wounds, or depending on a car, how fast a car might be going. Medium velocity, we've got 1 to 4 millimeter diameter, so a little bit larger droplets, and the velocity is moving much slower than medium velocity, or of the high velocity. And some examples might be like a beating or a stabbing. And then low velocity, much larger droplets here, 4 to 6 millimeters in diameter. And you can see here another visual representation. The velocity is about 5 feet per second, and this would be blunt object impact, like we said, with like a pipe. So where the difference lies between blunt object impact and beating, I'm not exactly sure, but I would imagine that with beating it would be more so repeated, a little bit faster. Blunt object, maybe like 1 or 2 like impacts themselves with something that maybe is a little bit heavier, so you can't go as quickly. I'm not sure. I've never conducted either of these injury patterns, so I couldn't tell you exactly the difference. But hey, this is why we're going to do a lab to see if we can create different types of velocity patterns too. This is another video that is linked on the folder there. This is actually really helpful when it comes to learning how luminol reacts with blood, so I very much recommend watching this video. Once again, this is the luminol video that I have linked in that folder. So when we are looking for blood, like I said, if you had watched that video, this should all be kind of a review. A perpetrator might try to remove blood evidence by cleaning an area, but that blood evidence is going to remain. It's very difficult to remove all of the residue. So even though a room might look perfectly clean, even if an entire cleaning crew went through, traces of blood might still be adhering to the surface even years after cleaning. So to detect hemoglobin, which is that, once again, protein that connects to the iron in our red blood cells, an investigator will mix luminol powder with hydrogen peroxide in a spray bottle. And it's then sprayed around to examine blood, and the iron from the hemoglobin actually speeds up this reaction with the luminol and the hydrogen peroxide, causing it to light up. So light is generated for about 30 seconds on the surface of the blood sample. Luminol can also be used to detect blood in darkened rooms or at night. And this is ideal for areas where the blood is not super visible. If you can obviously blood on a surface, then you don't need to use luminol. It's more so if you think that a crime might have occurred there, if you think there's any reason that blood might be present in this area, but it looks like it's been cleaned, that's when you want to use luminol, because then it will reveal to you the presence of the iron from the hemoglobin, which of course then correlates to the presence of blood. Now, if you find blood at a crime scene, it's not immediately eureka, hey, we did it, case closed. There's still some things that you have to do. You want to confirm that the stain is in fact blood. If it's not, then it's possible it could be ketchup, barbecue sauce. There was a case that we heard about the guy who was cleaning his shoes because they had barbecue sauce on them. The police, without even doing a test to confirm if it was blood, arrested him based off of those shoes. If they had just done step one to confirm the stain is blood, then maybe he would have never been arrested, right? And he's still in jail today. So confirming the stain is blood is important. You can use the Kastle-Meyer test. So if blood is present, a dark pink color will be produced. You could also do the leukomalachite green. So this undergoes a color change, producing green color if blood is in fact present. Now, the second thing you want to do is to confirm whether or not that blood is humid. That blood could be from a steak. It could be from something that somebody was hunting, if they were hunting deer, for example, from a chicken, from dog's food, if you feed them raw. There's so many different ways that blood could have arrived to that crime scene. So you want to confirm that the blood is in fact human. And so you can use what's known as an ELISA test, enzyme-linked immunoabsorbent assay, which essentially just uses an antibody protein that reacts using antibodies that then react to human blood proteins if they are indeed present. So as we see with blood type testing, which you guys will be doing in the other half of these blood notes, essentially what you do is you look for an antigen antibody response. So we're doing something very similar here in which we're just introducing antibodies to a solvent, and we're wondering if that solvent has human blood proteins or if there's other proteins present. So that will allow us to then determine whether or not the blood is human. And then finally, we want to determine the blood type. So blood typing is really helpful if you're trying to identify who that blood belongs to, whether it's for the suspect, whether it's being matched to the victim, whether you're trying to identify how many victims or how many individuals were at the scene. A lot of information you can get regarding blood type. So just as I had mentioned with the ELISA test, when we use antibody proteins to detect human blood proteins, we'll use the antigen antibody response as well to determine blood typing. So this is where we use a more specific antibody that's looking for either an A blood type antigen, a B blood type, or a positive or a negative, versus here when we're just looking for human proteins in general. So if no match is present, then a suspect may be excluded. You could also use blood for DNA analysis. You can use a lot of different factors that go into blood type. So there's lots of different ways that blood can help at a crime scene. And we're going to do a Science of the Media where we'll learn about a case in which blood was found at a crime scene. That blood was used to then arrest 2 individuals, but as it turns out, that blood was not human. So basically the entire basis for how these individuals were put in jail is based off of blood that wasn't even human. So we'll learn about that and we'll chat about the importance of it. But otherwise, these are all of our slides that we're going to be going through. I have these visuals here at the end of your slideshow, so you're definitely welcome to download them and look through them. I thought that this was a really good visual. I had to break it up into parts so it would all fit here, but it kind of just goes through everything that we learned, but has some really good examples, visual. Yeah, I just really like it. So this is here for you guys in your slides. If you'd like to continue looking through it, it's created by this website here. They have a lot of good information. But anyways, these are the slides. If you guys have any questions, let me know. Otherwise, I am excited to see how you transfer all of this information to that worksheet and once we do our blood typing lab in class. All right, thanks guys.